Hello, this is CJ, and in this video, we are going to talk about recombinant DNA technology or rDNA. This is much like an explanation of how transgenic organisms are produced. Okay, so recombinant DNA or rDNA technology, it refers to the process of joining DNA molecules from two different sources and then inserting them into a host organism to generate products for human use. So, basically, pag sasamahin mo nga yung dalawang DNA, tapos i-insert mo siya sa isang bagong host organism. Okay, hoping, of course, for a reason. So, to increase yield, o kaya para sa ibang mga reasons, gusto mo siyang palakihin, gusto mo siya maging color pink, ganon. So, that's the reason. So, those are the reasons, are an array of reasons. So, you don't just put DNA molecules inside to a host organism, okay? You don't just do that, tapos you just later on choose to cut and then join them, hindi ganun. So, you join them before you put them in the organism, okay? So, this process, it involves multiple steps and they must proceed in a specific sequence para makapag-generate ka ng isang desired product. The first step is the isolation of the genetic material. Alam naman natin that yung genetic material ng organism, ng lahat ng mga living organisms, they are nucleic acids. So, DNA or RNA. The first step in the recombinant DNA technology is i-isolate mo muna dapat yung gusto mong DNA in its purest form. So that is to say, dapat wala siyang ibang mga macromolecules. Kaso, sa isang normal na cell, yung DNA, hindi naman siya nag-exist within the cell membrane lang. Hindi naman dun lagi siya nakikita. It is also present along with other macromolecules. Hindi naman siya lang yung nakikita, okay, sa loob ng cell membrane. It is present along with other macromolecules such as RNA. Pwedeng meron din siyang kasamang polysaccharides, meron din siyang kasamang proteins, and lipids na hindi mo naman kailangan. So, yeah. How do we break open the cell and obtain the DNA that is free from other macromolecules? Pwede mong gamitin ang isa sa mga following na enzymes. Meron silang specific purpose since syempre enzymes nga sila. The first one is a lysozyme. To break the bacterial cell wall, kailangan mong gamitin lysozyme. And then, para na ma-break yung plant cell wall, cellulase. Kasi gawa yung cell wall sa cellulose. Para naman ma-break mo yung fungal cell wall, kailangan mo gamitin chitinase, chitin. Ayun. So, para naman ma-remove yung RNA, you use ribonuclease. And kapag protein naman, hulaan mo. Protease. Okay, so, yung mga proteins na kailangan mo matanggal ay yung mga histones that are associated with the DNA. And then, other macromolecules are removed, or you can remove them with their specific enzymes or through specific treatments. Ultimately, yung pag-add ng ethanol or ethyl alcohol, it causes the DNA to precipitate out as fine threads. So, this is then spooled out to give purified DNA. So, basically, the first step is to make sure that you have the pure genetic material, yung DNA lang. Okay, the second step is restriction enzyme digestion. Yung mga restriction enzymes, they act as molecular scissors. Sila yung pangkat ng DNA sa mga specific locations. Since yung DNA, it is a long strand, hindi mo naman kasi kailangan yung buong strand. So, kailangan mo siya i-cut. Yung isang area lang, yun lang dapat yung kukunin mo. And how do you cut it? Paano mo matatanggal yung mga hindi kailangan parts ng DNA? You use restriction enzymes. Yung mga reactions na to are called, yung mga reactions that involve restriction enzyme, you call them restriction enzyme digestions. They involve the incubation of the purified DNA with the selected restriction enzyme. Magkasama sila at conditions na optimal para sa pag-react ng specific enzyme na yon. Now, the technique agarose gel electrophoresis, it reveals the progress of the restriction enzyme digestion. This technique involves the running out of the DNA on an agarose gel. On the application of the kuryente, The negatively charged DNA, it will travel to the positive electrode and is separated out based sa size. This allows us to separate and cut out the digested DNA fragments. Now, the vector DNA is also processed using the same procedure. Now, what do we mean by the vector DNA? Ganito. Di ba, yung in-isolate mo dito is the DNA na gusto mo, na okay, yung in-isolate mo DNA dito, it belongs to that organism na mayroong characteristic na gusto mo. So, pagka-isolate ng DNA, ang ginawa mo is hinana mo yung specific na characteristic na yun dun sa DNA niya at ikakat mo siya ngayon. Okay? Ngayon, yung vector DNA is yung vector, yung uri ng DNA na gagamitin mo as tawag doon, tool para sakyan ng specific trait na kukunin mo nga dun sa isang organism. So, itong si vector, para siyang sasakyan. Okay? Ilalagay mo siya, yung specific trait na gusto mo, dito kay vector DNA, para ma-accept siya ng isang bagong organism. I hope you get that. Okay, so a vector DNA, it acts as the, parang means ng buong process para mahatid or ma-accept ng host cell yung trait na kukunin mo from another organism. Okay, DNA din siya pero ganun yung purpose niya. Okay, so the next one is the amplification of the DNA using polymerase chain reaction. Alam nyo na to, it's the method of making multiple copies of DNA sequences using the enzyme DNA polymerase. It helps to multiply or amplify a single copy or a few copies of the DNA into thousands or millions of copies. So, PCR reactions, they run on thermal cyclers using the following components. So, the first component, syempre yung template. Pangalawang component, yung primer. Anyway, I hope na you already know about polymerase chain reaction. Pero sige, kung sakaling hindi, ganito siya. Diba, sa restriction enzyme, digestion, kinat mo na yung DNA at kinuha mo na yung specific DNA fragment na gusto mong ipa-express sa isang bagong organism, tama ba? Ngayon, yung mismong DNA na yun, i-amplify mo yun, pararamihin mo yun. Gagawa ka ng maraming copies through polymerase chain reaction. Ang polymerase chain reaction, ginagamit niya yung DNA polymerase, it's an enzyme. Tapos, sinasubject niya yung DNA at saka yung DNA polymerase under different types of temperatures. Okay? Kaya nga, thermal cycler kasi sinacycle din niya yung temperature. Kasi, when you change the temperature, tapos may presence ng DNA polymerase, tapos meron din DNA, ang nangyayari doon is nagre-react yung DNA polymerase with the DNA. Minumultiply siya. How? Glad you asked. Mamaya, or for a separate video, we're going to talk about that. But for the meantime, the following are the needs. The first one is the template. Yung template is the DNA, yung kinat mo kanina from the restriction enzyme digestion. Pangalawa is the primer. The primer is a small, chemically synthesized oligonucleotides that are complementary to a region of the DNA. Alam niyo yung di ba, meron tayong adenine, meron tayong guanine, merong cytosine, merong thymine. Okay, so yung isa-isa sila. Halimbawa, yung basta, ang primer, it will take any of those nucleotides tapos magbabind siya kay DNA. Halimbawa, kung yung si DNA meron siyang C, si primer is going to be G. Ganun siya. So, magpe-pair up lang sila. Next one is the enzyme, the DNA polymerase, and then the last one is the nucleotide, which is needed to extend the primers, by the enzyme. So yeah, those are the nucleotides. And pero yeah, we'll talk more about the amplification. I mean polymerase chain reaction in a separate video. Pero for the meantime, I hope you get that in this process we're going to have multiple copies of that specific DNA sequence. We go now to the fourth step, which is the ligation of DNA molecules. So the purified DNA and the vector of interest, they're cut with the same restriction enzyme, pareho lang. Ngayon, since okay, meron na tayong na-cut na fragment of DNA at saka yung vector, open na siya, such as here, ilaligate mo lang yan sila. Okay, so pagsasamahin mo sila by the term ligation. So anong klaseng enzyme ang gagamitin mo? It's going to be DNA ligase. Again, ang gagamitin mong enzyme, DNA ligase. The resulting DNA is going to be recombinant DNA. In this example, yung green is the DNA, yung template kanina, o yung pini-CR mo. Tapos yung red is the vector. Kapag pinagsama mo yan, the vector and the DNA fragment together are called recombinant DNA. Okay. Next one. I-insert na yung recombinant DNA sa mismong host. So, sa step na to, yung recombinant DNA is introduced to a recipient host cell. Ang process na to is called transformation. Yung mga bacterial cells, usually, they don't accept foreign DNA easily. Kaya, kailangan i-treat muna sila para maging competent sila to accept DNA. So during transformation, if a recombinant DNA bearing a gene for ampicillin resistance is transferred to recipient E. coli cells, then the E. coli cells also become ampicillin resistant. Now, this aspect is useful in differentiating transformed cells from non-transformed cells. For example, if we spread the transformed cells on an agar plates containing ampicillin, only the transformed ampicillin-resistant cells will grow, while the untransformed cells will die. Therefore, in this case, the ampicillin resistance gene acts as the selectable marker. When you add recombinant DNA to a new organism, you are going to you are going to transform that organism. Okay, so I hope you get that process. So after ligating them, after joining them, you're going to insert it now to the host. Okay, so that now is going to cause changes to the host called transformation. Paulit-ulit, sana magets nyo. Okay, next one is obtaining the foreign gene product. The gene product is not the recombinant DNA. Okay, so the recombinant DNA multiplies in the host and is expressed as a protein. Now, under optimal conditions, this is now the recombinant protein. Now, small volumes of cell cultures will not yield a large amount of recombinant protein. Therefore, yung mga large-scale production is necessary to generate products that benefit humans. For this purpose, vessels called bioreactors are used. Yung recombinant DNA kanina, hindi mo pa naman yun kinikilala as the gene product. Okay, so yung halimbawa na-insert mo na siya, yung mismong recombinant protein yan, ang gagamitin din para makapag-generate ka ng mismong gene product. The recombinant DNA is going to produce recombinant protein. Wag na natin, scrap the part where I say hindi siya yung gene product. Okay. Okay, so yeah, again, obtaining the gene product, the recombinant DNA is going to produce or to signal the production of a recombinant protein. Ayun siya. Now, to make sure that we have enough of these recombinant proteins, bioreactors which are large containers as shown are employed. The use of them is employed. So it is where the fresh medium is added from one side and used medium is taken out from another side. So these bioreactors can process around 100 to 1,000 liters of cell cultures. They provide the optimum conditions such as temperature, yung oxygen levels, yung pH levels, and even vitamins. Okay, they are providing all of them to biologically convert raw materials into specific proteins or enzymes. Now, a stirred tank bioreactor, which is the common type of bioreactor, is usually cylindrical and has the following parts. So, yung mga ibang parts na sasabihin ko, perhaps wala dito sa illustration. Pero one of them is the agitator system para nag-i-stir, okay, patuloy na gumagalaw yung contents. Tapos, yung oxygen delivery system para yung air, okay, yung air input, ayun siya. It's continuous. Tapos, meron siyang foam control system. It's a foaming aerator, yung submerged one, ayan. And then yung temperature control system, meron tayong thermal jacket dyan, okay? And then pH control system, it can actually be one of those sensors. And then meron tayong sampling port. Okay, so sampling port. Okay, so now for the last step in all of these for the production of transgenic organisms, the last one is downstream processing. So before the protein is marketed as a final product, it is subjected to downstream processing that includes separation and purification, formulation with suitable preservatives, clinical trials to test the efficacy, which is really one of the most important things that must happen actually, and then safety of the product, and then quality control tests. Ayun. So I hope that you were able to get the most important the most important parts of this video, of this topic, with the help of this video. So do not forget to always keep in mind these basic steps in the production of transgenic organisms. This is AJ, and it's already 12:32 a.m. from where I am. Alright, so if you're watching this video on the same time, manginig ka, char. Alright, so good night, everyone. Goodbye. This is the Knowledge Catalog Education Solutions.